Hello, senior science students. This video is to help you answer question 4 for your senior projects. It requires a flash experiment. Recommend you go through this yourself. If you have access to a laptop or a desktop computer, it'll be easier to answer those questions. But for those of you who do not have access to a laptop or a desktop, instead of doing the flash simulation or going through the exercise yourself, we're going to go through it together in this video. Alright, so in this online simulation, the intro picture says, you are holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? So on the screen we're seeing DNA strands. So DNA is a double helix, two strands that are coiled together. If they are cut up, they can be of various different lengths. Here they're showing short versus medium versus long strands. If the DNA strands were as big as your shoelaces, you could sort them by hand into groups and measure them. But DNA strands are molecules so tiny that you can't see them even under most microscopes. Is there a way to sort and measure the DNA strands in your tube, even though you can't see or touch them? There is. It's called gel electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules, like proteins. How does this work? The gel, shown in the magnifying glass here, is the filter that sorts the DNA strands. It looks like a sponge made of jello with many small holes in it. So we just placed DNA samples into those wells at the top of the gel. So next we are adding a current that's going to be running through the gel as well. Electrophoresis is how we push the DNA strands through the gel filter. By adding an electrical current, we can make the DNA move. Okay? So the DNA has a charge on it, and then the current is going to help the DNA repel from the side of the well, the negative side that it's on, and it's going to be attracted to the positive side, and the DNA is going to move through the gel from the negative side that it starts towards the positive side. And this is a little image showing how the DNA is going to move through the gel. Short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, DNA strands in the sample sort themselves. So you can think of this as a race. So all the DNA strands are starting at the same time, but the shorter ones are going to be lighter and faster. They're going to be able to move through the gel the quickest. And the longer sequences are larger and slower, and they're going to move down the gel last. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of strained DNA strands together. These groups show up as a band in the gel, as you can see here. So each of these spots isn't one DNA molecule, but a bunch of DNA molecules of the same size grouped together. Now it's your turn to run a gel.
Interesting.
That's the scientific slang for gel electrophoresis. Follow along with the steps shown above. So first, we're going to be making a gel. We're going to set up the gel apparatus. We're going to load the DNA sample into the gel. We're going to hook up the electrical current and run the gel. And then finally, we're going to stain the gel and analyze the results. We're going to do that whole thing in this simulation. This video will likely be broken up into parts. Make sure to watch each part to fill out the activity questions and then submit your final assignment for the senior project. Alright, so now we are in a simulated biology laboratory. And here's what we'll need to make a gel. We have powdered agarose, buffer, a flask, a microwave, a gel mold, and the gel comb. Making an electrophoresis gel is a lot like making Jell-O, but you don't want to eat this type of gel. Put a small amount of agarose into the flask. Agarose is a dry powder similar to gelatin but made from seaweed. Add some liquid buffer to the flask. The buffer is a saltwater solution that will let electrical charges flow through the gel. We've loosely placed plastic wrap at the top, as you can see here, over the top of the flask to prevent the liquid from boiling over. Open the microwave by pressing the open button and place the flask containing the buffer agarose mixture inside the microwave. Heat the mixture until the agarose melts into the buffer. And now this is all melted together. We've removed the plastic wrap from the top of the flask. Pour the melted agarose mixture into the mold. Notice that the mold has tape on each end to hold the melted agarose. Okay, so we don't want to be spilling out. So we're going to pour this into the mold. Place the comb into the gel at one end. The notches in that gel mold will make the wells. Let the gel cool and solidify. This usually takes about half an hour, but we'll speed up the clock for you. As the gel cools, the tiny holes will form in it. Carefully remove the comb, leaving empty wells for the DNA samples. Now your gel is ready to run. Now it's time to set up the electrophoresis.